All right, everybody, welcome. And what I'm going to be talking about today will be the Calvin cycle. Just kind of a brief overview of the Calvin cycle, not extreme detail here, but I want to explain some of the main parts of the Calvin cycle. And before we get started, I want to talk about the goal of the Calvin cycle, and that is to take up CO2, carbon dioxide, and generate a molecule called G3P, or that's our sugar molecule that I've been talking about in class. So in doing this, there are three sort of stages here. We're going to say the first stage, stage one, is fixation, where we are going to fix the carbon from CO2 into a carbohydrate. And then the second stage is called reduction. So remember oil rig or redox reaction. So in this particular case, we are going to reduce the molecule at the end here, or toward the end. And then three, we are going to regenerate our starting material. Regenerate or regeneration. Because of course this is called the Calvin cycle, right? So it's a cycle. So we're going to regenerate our original starting material. All right, so let's dive into it. Only a few names of molecules you need to memorize here, but one of them, fairly easy. We're going to go ahead and start with 3 carbon dioxide molecules, and that's going to be our input from the atmosphere. So in through the stomata of the leaves of the plant, we're going to pull in some CO2, and then that's going to be combined with another molecule. That other molecule is going to be 3 molecules of, it's called ribulose bisphosphate, and that's a mouthful. The abbreviation for that is RUBP, and I'm okay with the abbreviation. So in this particular case, ribulose bisphosphate, there are 3 of these and 3 of these. You don't really need to memorize, at least for my class, the numbers of all these like you did in cellular respiration. But importantly, these molecules are going to be combined, or at least the carbons of them, are going to be combined, and that's going to be done with the help of an enzyme called Rubisco. And that is thought to be one of the most common proteins on planet Earth. So it's a really important molecule. And what we're going to generate from that is what we call an intermediate molecule. And it's short-lived, intermediate molecule. And the important thing I just want to point out here is that this is a 6-carbon molecule. And it's short-lived. What's going to happen is we're going to break that sort of in half, and we're going to then generate a 3-carbon molecule. You don't need to know the name of it. I'm just going to draw it as little carbons like so. So a 3-carbon intermediate in this particular case, and this is equal to carbon. Okay, so hopefully that will help us understand this image, and we'll go forth with that as an idea. And yes, that has a name. For those of you that would like to know, it's 3-phosphoglycerate. But in our for our means, we just need to understand that we are going to break it down into a 3-carbon molecule. All right. Okay, so what is all this, right? Well, what we're actually talking about with all this is, I'll use a different color here, blue. So what we're seeing here is something we call fixation. So all of this part right in here is called fixation, or carbon fixation. All right, that's fixation. Okay, so let's continue on now. We're not done. We've just taken carbon and made it, put it into this other molecule here. It's a 3-carbon intermediate sort of molecule. Now let's go ahead and continue on with this guy. We're going to have 2 main things happen here. We're going to first incorporate ATP into our molecule. Well, the phosphate from ATP, I should say. So in this particular case, ATP is used to sort of couple this next reaction. And by the way, this stage, just because you know where we're going probably, is the reduction. We just covered fixation, that's up here. The reduction stage is next. This isn't reduction per se. What it is going to add a phosphate group to our molecule. So this is our molecule. We'll go ahead and redraw it here. Here are the carbons. And then we go ahead and we add our phosphate group on the end, like we did here. But now we're going to grab another phosphate from that triphosphate and put it here. All right. Okay. So that's what our molecule looks like now. And again, remember back to ATP coupling, the energy coupling that we discussed previously. So in this particular case, this is the reduction stage. So NADPH, we're going to have a redox reaction. So what we're going to end up with is NADP+. This is a reduced molecule. This is an oxidized molecule. We're going to have this redox reaction where the electron gets passed. In this particular case, we also lose a phosphate group, so we're going to lose this phosphate group here. All right, importantly, this stage, all this stage here that's going on, this is called the reduction stage, and really important here, we're going to produce G3P. This molecule is called glyceraldehyde, glyceraldehyde 3-phosphate. I'll abbreviate it, okay? And that is a 3-carbon molecule, just like it was before. It's a little bit different, but in this particular case, we'll just kind of draw what we see. Okay, I'm leaving my, there we go. Lost my pen for a second. We'll draw this image, but we've lost that phosphate group, so in this particular case, that's what it looks like. Now, what you need to understand is this entire process right here was regeneration, so we'll go ahead and we'll put a box around this section right here. Reduction. I said regeneration. This is reduction. So reduction. Alright, that's the reduction stage. And that's great. What we've done is we've made our G3P. Now, one of these will go on to give us glucose, and I'll just put other molecules. Okay, lots of other stuff. Now only one of them. In this particular case, we have 6 total. So 1 goes that direction, and then 5 go on this direction. And some things happen here in which we're going to take our 3-carbon molecules, and ultimately we're going to make this RUBP again. This is our regeneration stage. So we're going to regenerate that RUBP, and that's going to require ATP. So ATP, ADP, and guess where that phosphate goes? Yep, you got it. It goes to the ribulose bisphosphate. So we've regenerated our RUBP. This again is our regeneration stage, so go ahead and write regeneration. Right, regeneration stage, so that's this part over here. Right here, okay? So regeneration stage there. So follow along just as we kind of review. There are the main goal here is to fix carbon from carbon dioxide in the atmosphere into G3P. You see that over here. And then that's our main goal, if you will. We're going to then do this through what we call the Calvin cycle, which comprises of 3 steps: fixation, reduction, and regeneration. And as we walk our way through it, we can see some of the key components here. Carbon from carbon dioxide is added to ribulose bisphosphate, or RUBP, with the help of an enzyme called Rubisco. And then that, through a couple of steps, forms this 3-carbon intermediate. Actually goes by the name of PGA. In this particular case, we have finished our fixation step. We now proceed to reduction, in which we incorporate a phosphate from the ATP. We see that here. And then we are going to reduce, and in this particular case, we reduce our molecule by with that electron from NADPH, and in the process we lose NADP+, and phosphate group. These then go back to the light reactions where they sort of come back as NADPH and ATP. Finally, we get our G3P. One of those, G3P, goes on to help us make glucose. The other 5 are then used to regenerate our RUBP. That process, just so you know, will require some energy input, ATP, as we see here. So fixation, reduction, regeneration, with the main goal of incorporating the carbons from CO2 into the carbons of glyceraldehyde 3-phosphate. All right, I hope this helps, and I'll see you in class. All right, talk to you later.